Talk about the integumentary system, and the integumentary system is composed of your skin, hair, nails, and exocrine glands. So exocrine glands, exo to the outside, crine means secretion, and so exocrine glands mean that their secretions go to the outside. So let's talk about some overall functions of these. Skin, it is literally a barrier between the external environment and the internal environment. And because of the composition of our skin, it protects us from chemical damage to a certain extent, environmental damage, UV damage, as well as disease. So we know that you already know that the outermost layer of the skin is stratified squamous epithelium. So having lots and lots of layers of those squamous cells means that there's a thicker layer between what is outside and what is inside, so harder for certain bacteria and other things to get through. Hair, think of hair, thermal, helps with regulating body temperature. Nails, these are going to be more of a protective function, like against predators. You can scratch someone if you feel threatened. You can also use nails as tools to pry apart things. And then exocrine glands, their secretions go to the outside. And the main ones we're going to talk about are sebaceous glands, which are oil-producing glands. Oil meaning it's a higher lipid content, so more mucousy or thicker, more viscous. And so sebaceous glands, the oil-producing glands, this oil goes to the outside and it helps lubricate and moisten our skin. We know that if our skin is not moistened, it is more dry, more susceptible to cracking and breakage. And if your skin cracks and breaks, you have open wounds, and this means that you could potentially have a bacterial infection. So we want our skin to be moist and lubricated. Eccrine sweat glands, these produce more of a watery substance. So their watery substance goes to the outside, and the main function of eccrine sweat glands is going to be for evaporative cooling. So the water gets released to the outside, your body is overheated, that water evaporates off of your skin, pulling heat with it. So eccrine sweat glands are important in evaporative cooling. So the skin has layers, and the epidermis, so epi meaning above, dermis, above the dermis. This is the most superficial layer. And you already know what tissue makes up the epidermis. It's stratified squamous epithelium. So this is where learning all those tissues and learning their locations and functions helps you throughout the rest of Anatomy 1 and Anatomy 2. So epidermis is made up of stratified squamous epithelium, lots of layers of squamous cells. The majority of the cells are keratinocytes. So cyte meaning cell, keratino pertaining to keratin, which is a waterproof protein. So keratinocytes make up the majority of the cells. Next we have melanocytes, which are also found in the epidermis. So melanocytes, melanin producing cells. Okay, so melanin is a pigment. And the definition of a pigment is it has the ability to absorb UV light. And so melanocytes produce melanin, and that pigment absorbs UV radiation, so it protects us from sun damage. Langerhans cells, these are thought to be important in like an immune response, but they're a very small minority of the population of cells of the epidermis, the majority being keratinocytes. These are sublayers within the epidermis, and stratum meaning layers, and I'll talk about these specific layers in just a second. The dermis, which is underneath the epidermis, you already know what tissue is found in the dermis. Again, you learned this for the exam 1. The dermis is made up of dense, irregular connective tissue. So remember that densely compacted collagen and elastic fibers, majority being collagen fibers. They're running in all different directions. That's where the irregular part comes in. Collagen is important. It's a very strong protein fiber that is really good at resisting pulling forces. It has high tensile strength. So The dermis, being dense, irregular connective tissue, means that it resists pulling forces in all different directions. You can think all day as you're moving and your skin gets pulled in all different directions. Well, we don't want that pulling to make the skin, essentially saggy, right? So we have lots of collagen, which resists pulling forces. Okay? As you get older and when you get wrinkles, that collagen is breaking down, no longer able to resist pulling forces as well. You also find, in connective tissues, you find nervous tissue and vascular tissue as well. This is something that is unique to connective tissues. So I'll talk about some of the nervous tissue that you find in the dermis, and you'll also see it's very highly vascularized. Okay? Remember, epithelium is not vascularized. So the epidermis is made up of stratified squamous epithelium. It is not vascularized, but there's still tons of cells. They're going to get their nutrients from the blood vessels that are found in the dermis, because the dermis is very highly vascularized. Okay? There are layers of the dermis, and I'll talk about these two layers in just a second. Below the dermis is the hypodermis, so hypo meaning below. Hypodermis is the deepest layer. You already know what tissue is found in the hypodermis because you had to learn locations of loose areolar and adipose. So loose areolar connective tissue and adipose make up the bulk of the hypodermis. This is the layer that connects the epidermis and dermis to the muscles below. So if I were to add another layer below this, below the hypodermis, it would be all of your muscles. So the loose areolar connective tissue, and embedded within that loose areolar connective tissue is lots and lots of adipose. That all connects the epidermis, dermis to the muscles below. Because it's lots of loose areolar connective tissue and lots of fat, it also adds as a cushion, a protective cushion. Okay, so we don't have as much force being applied onto our muscles, so it provides cushion and protection that way. Adipose, those adipocytes, remember I talked about how they store lots and lots of triglycerides, so they're also an energy storage tissue, so that adipose connective tissue can serve as energy storage. It also, the hypodermis, another function of the hypodermis is for temperature regulation. So if our body temperature drops too low, we can start to break down those triglycerides in the adipose, right? We can break down those triglycerides, generating heat while doing that, and that heat gets distributed around our body. Okay, so the hypodermis, those are all functions of the hypodermis. Okay. All right, so now let's talk about the specific layers within the epidermis. So again, this should be repetition as far as identifying, because you've already seen this. This is the epidermis. This is stratified squamous epithelium. Lots and lots of layers of squished squamous cells. You find stratified squamous epithelium anywhere that's open to the outside and subject to abrasion. So you have lots of layers of cells in this epithelium. So the top layers can slough off, and we have layers below. Our skin would be really crappy if our skin was made up of simple squamous epithelium, because that's only one layer. We'd be leaking everywhere, and you'd only have one layer. So if you scratched it off, that's it, right? So having multiple layers allow anywhere where it's open to the outside, subject to abrasion, which our skin is literally the most outside organ that we have. The skin is considered an organ, okay? It's the largest organ of the body. Okay, so, and this is, you remember me showing you in, for exam 1 material with the tissues, we have stratified squamous epithelium, and sometimes you'll see this big, thick, dead layer, okay? This is all dead cells, okay? Just forms even more of a protective barrier between the outside and inside. So let's talk about the layers and what's happening. Well, I'm going to start from the deepest layer to the most superficial. So the deepest layer, which is going to be, look, so you can tell this is stratified squamous epithelium, and then it dramatically changes to all of this connective tissue below. So the deepest layer of the epidermis is going to be that layer which is right next to the dermis below. And these cells are somewhat they look almost cuboidal in nature. Not exactly cuboidal, but they don't have that really nice squamous shape to them. That's because these are the cells of the stratum basale. Basale meaning basal most, bottom most, okay? So the stratum basale cells, these are the cells that are undergoing mitosis constantly. So mitosis generating new identical cells, right? Making copies of themselves. So these cells are constantly undergoing mitosis, so that's why they look larger. They're not as squamous. Why do they need to undergo mitosis constantly? Well, because the cells up at the top slough off and we have to regenerate more. Slough off, regenerate more. So the stratum basale's function is to undergo mitosis and generate more cells for the epidermis. Now, you have to be able to identify these layers on a microscope slide. So if I have the pointer within the epidermis, because it's stratified squamous epithelium, and I have the pointer right here at the very bottom layer, right next to the dermis below, you say stratum basale of the epidermis, okay? Now, I'm going to jump ahead to this layer right here where you see it's much darker. This is the stratum granulosum. Stratum granulosum. Okay, this is where all of those keratinocytes, okay, keratinocytes are the main cell type of the epidermis. This is where they are completely full with keratin granules. Okay, that's why they have that such a dark appearance. All right, now the bottommost layer, easy to identify because it's right next to the dermis. This is the stratum basale. Okay, mitotic divisions generating more cells. In between the stratum basale and the stratum granulosum, all of these cells that are just regular old squamous keratinocytes, okay, they're just squamous cells, this is the stratum spinosum, okay, this is the stratum spinosum. So if I have the pointer right here and it's in between the stratum basale and the stratum granulosum, you say stratum spinosum of the epidermis. These are keratinocytes that have just been generated and they're starting to fill up with keratin granules. Keratin is a strong waterproof protein. So, stratum basale generates new cells, new keratinocytes. Those keratinocytes move up through the stratum spinosum and they start to fill with keratin. When they get to the stratum granulosum, they are so full with keratin, right, that they don't have room for anything else in the cell. So they can't undergo regular metabolic reactions. So they're so full with keratin that they start to die, okay? So this dark layer right here, the stratum granulosum, these are keratinocytes that are completely full with keratin. Then they die. They undergo apoptosis because they're so full with keratin, they can't do anything else. Stratum lucidum. You don't have to identify this on a microscope slide, but I wanted to show you a picture that has the stratum lucidum. This is that slightly lighter layer right above the granulosum, and it's called the lucidum because it looks very transparent, and this is where the cells have popped, undergone apoptosis. Okay, so it looks somewhat translucent. It's really hard to see on the slides that we have, so you don't have to identify it. Don't worry about it. But you do need to be able to identify stratum basale. These are the cells right next to the dermis. They're undergoing mitotic divisions, generating new keratinocytes. Those keratinocytes move up through the stratum spinosum, starting to fill with keratin, move up even further to the stratum granulosum, this dark layer. They're completely full with keratin granules, and they undergo apoptosis and die. Then they move up to this big, thick, dead layer, which is the stratum corneum. Okay, when you think of like a corn on your feet, or right, like a stratum corneum, this is, and you can tell, you see that they're all dead because you don't see nuclei anymore. There are no more nuclei. So these are just really densely compacted dead keratinocytes, okay? And it just forms a much thicker protective barrier. You find this thick, so how thick the stratum corneum is depends on how much abrasion that area of the skin is subjected to. So you know, like if you work with your hands a lot, you'll have calluses. Well, where you have calluses, you have thicker stratum corneum. okay? Palms of your hands, soles of your feet, okay? Those are areas where you'll have a thicker stratum corneum, okay? Let's see. Fun fact, so in humans, the stratified squamous epithelium of the esophagus is never keratinized. Like you don't have a thick stratum corneum. You don't have a stratum corneum at all because we eat soft foods. But if you were to look at the esophagus of, say, a squirrel, which has a nut-heavy diet and nuts are very abrasive, their esophagus actually will have a little bit of the stratum corneum. Kind of fun fact for you. Okay, so Stratum Basale, Stratum Spinosum, Stratum Granulosum, Stratum Corneum. You should be able to identify those layers on a microscope slide. You'll notice that the Stratum Basale is very, the epidermis altogether is very highly invaginated, right? It's invaginated because what do invaginations do? It increases the surface area, right? Having these dips into the dermis means that we have increased surface area of the stratum basale, meaning we have more cells that can undergo mitosis, right? These portions of dermis that jut up into the epidermis, these are called dermal papillae, okay? Dermal papillae. This is what gives you your fingerprints. So, and why fingerprints, so it's your dermal papillae that gives you your fingerprints, and each person's fingerprints are unique to them because the way that you move around and touch inside of your mother's uterus during embryonic development, I'm going to touch and move around differently than you are. And so that causes differences in where these invaginations are, okay? And so these are called dermal papillae, okay? Dermal papillae. Highly invaginated stratum basale, meaning more cells that can undergo mitosis. This is an image, a cartoon image of the cell, or the skin, and you should be able to identify, I have a document posted, and you should be able to identify the structures and know the functions of, if your question ever is, do I need to know the function? Yes, because it's anatomy and physiology. You have to be able to identify the structure, and you have to know a function. That's the physiology part, is knowing functions. So these are like the models, the plastic models that we have in lab. So some superficial most layer, this is your epidermis. Okay, this is your epidermis. You can even see the highly invaginated nature. You can see, so everything from the epidermis down, this is all dermis. Okay, it's a pretty thick layer, and you see lots of stuff embedded in the dermis. Remember, the dermis is where you find nervous tissue and vascular tissue. These are all blood vessels. Down here at the bottommost, these yellow large cells, these represent adipose. So where you start to see lots of adipose, this is the hypodermis. Hypodermis, adipose. Dermis, dense irregular connective tissue. Epidermis, stratified squamous epithelium, okay? Some things that you have to be able to identify within this. Let's see, I've already talked about the dermal papillae, okay? Let's see, hair shaft. Okay, so these are your hair shafts, right? And again, remember the function of hair, thermal regulation, body temperature regulation, as well as UV protection. So hair shafts, erector pili muscles. So this right here, this is an erector pili muscle, and exactly what it sounds like, it erects the hair shaft. So this is a muscle under involuntary control, and when it contracts, it pulls on the hair follicle and it erects the hair shaft. This happens, this is because it's involuntary, it's also under emotional control. So if you're frightened or, scared, then this contracts. And think about like lower animals who are more furry and have more hair. If you're scared, if a predator comes to you, okay, and you get scared and you want to fend off that predator, you want to look bigger. So these erector pili muscles erect the hair shafts, and that animal can appear bigger, right? And hopefully scare off any predators. It's also thought that this helps with temperature regulation. So when you're really cold, you'll notice you get your goosebumps. Those goosebumps are your hairs erecting. So you get cold, your erector pili muscle contracts. It erects the hair shaft, and what that allows is it allows air to get trapped within these hairs, and so warm air stays near the body. So it also helps with temperature regulation as well. Let's talk about some nervous tissue that you find. Meissner's corpuscles versus Pacinian corpuscles. So Meissner's corpuscles, these are little nervous receptors that are there's up in these dermal papillae, right? So here's a Meissner's corpuscle. They're shoved up in the dermal papillae. So that means they're really close to the surface of the skin. The dermal papillae is right underneath the epidermis. So these are shallow touch receptors or light touch receptors. So Meissner's corpuscles, this nervous tissue has the ability to detect really light touch. Makes sense because it's really close to the epidermis. So lighter touch would be detected by these, okay? So light touch, when someone just grazes your skin with their fingertips, it's your Meissner's corpuscles that are picking up on those vibrations. Pacinian corpuscles, look, they're huge and they're located really deep within the dermis. So they're almost to the hypodermis, right? So Pacinian corpuscles are deep touch receptors, meaning that it takes larger vibrations in order to stimulate these Pacinian corpuscles. Okay, so these are deep touch receptors. So when you really push on your skin, you're getting a deep tissue massage, right? These Pacinian corpuscles are picking up on those vibrations. So this is, again, that structure-function relationship and where things are located and why the location and structure is ideal for the function it carries out. Meissner corpuscles, more superficial, lighter touch receptors. Pacinian corpuscles, deeper, deep touch receptors, okay? Structure-function relationships, always know them. Free nerve endings. So free nerve endings, and usually in models, nervous tissue, as I say usually is colored yellow. Free nerve endings, so you can see that these little tiny endings of these sensory neurons will actually go up and innervate into the epidermis. So they're really superficial. They have endings within the epidermis. Free nerve endings are important for pain and temperature detection. Totally makes sense, right? Because if something is painful, you want to detect it really early on. If something is extremely hot or extremely cold, you want to detect that really soon. So these free nerve endings are located way up in the epidermis. They stick up into the epidermis, right? We wouldn't want pain receptors and thermoreceptors all the way down in the dermis, because that means that if it's, something that's cutting, it would have to cut all the way through in order to be detected down here. So pain receptors and thermoreceptors are these free nerve endings that go up into the epidermis, okay? Sebaceous glands, again, those are the oil-producing glands. They produce that, that the substance, their secretions are high lipid content. Sebaceous glands have an alveolar shape to them. Alveolar means like a grape-like shape. They're always associated with a hair follicle. So here's your hair shaft. Here's your hair follicle. These very bubbly-like glands, okay? Grape-like glands that are directly attached to a hair follicle, these are sebaceous glands, okay? Sebaceous glands are always associated with a hair follicle. So that those oil secretions get dumped into the hair follicle, move its way up, and exit through the same pore that your hair shaft exits through. So it lubricates your hair. We all know that because if you don't wash your hair for a long time, your hair becomes a little oily, but it also moistens and lubricates the skin. Sebaceous glands. Equine sweat glands are coiled. So these guys right here, they're very highly coiled. They're tubular in shape. These are alveolar in shape. Tubular in shape, right? So they're very highly coiled, and they have this single tubule okay, this single tubule that leads to a pore, okay, leads to a pore. And its secretion will be that watery substance, which again, elevated temperature, equine sweat glands produces a watery substance, goes out through this pore. Then we have it on our skin, and then hopefully if the environment isn't super humid, then that water will evaporate, pulling the hot air with it. Since we live in South Louisiana, we know that sometimes the air is like water because it's so humid, and so that evaporative cooling doesn't work as well because the water just stays on our skin, okay? But hopefully it's not super humid and that water can evaporate and cool us off. Let's see, I think that's it. And then again, you'll see arteries denoted in red, veins denoted in blue. Again, this is all the vascular tissue, and you see that it runs all the way up. This vascular tissue runs all the way up to right underneath the epidermis. And that's another point that I want to make, is that these cells, okay, so the cells of the epidermis, these are not vascularized. Epithelial tissues are not vascularized. They have to rely on diffusion of nutrients from the blood vessels in the dermis. Those nutrients have to diffuse from those blood vessels up to the cells of the epidermis, okay? So the cells of the stratum basale are going to receive the most nutrients. Perfect, because what are they doing? They're undergoing lots of mitotic divisions, so they need lots of nutrients, right? What happens when we move up to the stratum spinosum? They're filling with keratin. They're also getting further away from the nutrient supply, right? So nutrients have to diffuse all the way through the stratum basale cells and into the stratum spinosum. So these guys are getting fewer nutrients. They're also filling with keratin. Then those keratinocytes continue to move up and be pushed up because more cells are being generated down here. It pushes them up, and then they become the stratum granulosum. They're super full with keratin. That's why they're dark in appearance. They're also really far away from the nutrient supply of the dermis. So they're full of keratin. They're also really far away from the blood vessel supply, the nutrient supply. So guess what? They're really gonna die. So as these cells move up and get further away from the nutrient supply of the dermis, it also causes them to undergo apoptosis and form this dead stratum corneum. Okay. Here is a slide, histology slide of skin on low magnification. So you can literally see all three layers. You can see, look, where it's very highly invaginated, this darker pink, and this big dead stratum corneum. This is all epidermis, right? All of this tissue that's in the dermal papillae, all the way down to, oh, what do you see down here? Lots of adipose. So from here to here, this is all dermis. You can see that densely compacted collagen fibers running in all different directions, dense irregular connective tissue. And then once you see lots of adipose, this tells you you're in the hypodermis because it's primarily adipose and loose areolar connective tissue. So stratified squamous epithelium of the epidermis, dense irregular connective tissue of the dermis, and adipose connective tissue of the hypodermis, okay? Layers of the dermis. You have the papillary layer and you have the reticular layer. If I were to draw an imaginary dotted line where the lowest points of the epidermis are, okay, so a dotted line across here, all of that connective tissue that's shoved up in the dermal papillae, that's called the papillary layer. So if I have the pointer in a dermal papillae and I ask, what is the specific layer? You tell me papillary layer of the dermis. If I say, what is this structure? You say dermal papillae, okay? From where those dermal papillae stop, all the way down to where, until the hypodermis begins, that is all called the reticular layer of the dermis. So if I have the pointer anywhere that's not in the dermal papillae, but it's still all of that dense irregular connective tissue, and I ask, what is the specific layer? You would tell me, reticular layer of the dermis. If I have the pointer down here and you see lots of adipose, and I ask, what is the specific layer? You tell me hypodermis. Okay? I can also ask, I could put a pointer here, 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 or here, and I could ask you, what is this specific layer? You would tell me stratum basale, stratum spinosum, stratum granulosum, or stratum corneum of the epidermis. Okay? If I have a pointer here, this is in the connective tissue of the dermal papillae, and I say, what is the specific layer? You tell me papillary layer of the dermis. Okay? This is all stuff that I will ask you, and it will be on the test, so know it.